Today, I'll be presenting a case study from Dr. Ferreira's lab from D.A.J. So I would like everyone to meet D.A.J. D.A.J. is a patient of Dr. Ferreira's who has been diagnosed with a pituitary adenoma, a GH secreting one, as well as an insulinoma. Oh, sorry. Okay, so his tumors reflect the symptomology of MEN1, multiple endocrine neoplasia type 1, and that is a genetic condition that is associated with endocrine tumors. So D.A.J. Has a pituitary adenoma and an insulinoma, but when he was screened for the MEN1 mutation, it was not found in his germline, which means he does not have it. It can only be genetically passed down. So because of that, the Ferreira lab questions if the D.A.J. has mosaicism. It's not moving forward. Okay, yeah. No, that should be good. So sorry. Yeah, so the Ferreira lab hypothesizes that D.A.J. has mosaicism instead of MEN1 because mosaicism creates differentiated cells around the body that can create their own structures. So to find this, to test the hypothesis, the Ferreira lab does a process called whole exome sequencing via Northwest Genome. And before sequencing, the samples go through different steps to get the final result of a library, an exome library, and then the exome library is sequenced afterwards. After the experiment, we move on to data analysis. And I know this looks very complicated, but we're going to break it down. Yeah, so first, the data goes through a quality check before pre-processing. And after pre-processing, the reads are aligned to a reference genome and converted to a BAM file, stands for binary alignment map. Those files then go through another quality check step called base score quality recalibration. And after recalibration, variant calling takes place to find the somatic mutations in D.A.J.'s tumor, excluding the germline because we know that it wasn't genetically inherited. And the variants are then annotated using public databases before they are filtered and prioritized, which will give us the list of the most damaging mutations that are in the two tumors, the adenoma and insulinoma. Okay, so for results, this is an example of how we view the data. So on the leftmost column are the total list of mutations found in both tumors. The next column next to that is the mutations that were found in common between both the insulinoma and the adenoma. And then you see that on the right side, the most right is the adenoma and the middle is the insulinoma. So we found, we looked through this data and we categorize all the different variants as shown there. And we found that there were 4 common mutations between both the insulinoma and the adenoma, being TDJ, NCOR2, LTBP3, and MBFP12. So to break it down a little bit further, we found that 66 of the variants were found in the insulinoma, 30 of them were found in the pituitary adenoma, and then the 4 mutations were the remaining out of the total 100. And they had a common average allele frequency of about 3%. Oh, the slide was skipped. It was the, yeah. I'm not too, okay, I guess I can tell you there's no slide for it. There was, I'm so sorry. But because of the low allele frequencies of the mutations, we confirmed that D.A.J. is a mosaic because the allele frequency in these 4 common mutations were 3% versus the other mutations that were found in the insulinoma and adenoma, the allele frequencies were around 20%. So because of that big range of difference, we basically think that he's a mosaic, but we can't confirm that until we do further validation sequencing through bench work, comparing these tumors to other of the same tumors in different patients. And then when we did a string analysis, it's basically when you compare, you put genes into a database and it shows how they are connected. So we put the 4 mutations there and we found no direct mutation, no direct connection, but there was an indirect connection via a different gene, SUMO1. It stands for small ubiquitin-like modifier 1. And there's a connection between TDJ and NCOR2. And we think that because of that, ubiquitination might be a factor, and how the tumors are kind of, and the mosaicism specifically in D.A.J is kind of disrupting how he's able to live in the symptomology. So yeah, sorry for the hiccup, but I'd like to thank everyone who made this program possible and successful. I've learned so much and I really enjoyed being here this summer. And that's all due to everyone, their dedication, their kindness. Like, I've really enjoyed myself. And I'd like to thank the audience for listening.